Meiosis is a reduction-division process that contributes to genetic variety and makes sperm and egg cells, otherwise known as gametes. Majority of the cell cycle is spent in interphase, a phase in which a cell performs its normal functions while growing. Interphase can be split into three subphases: G1 phase, first gap; S phase, the most important phase of interphase, in which DNA is synthesized, chromosomes are duplicated, and cytoplasmic cells are doubled; and G2, second gap. During all three subphases, the cell grows. In interphase, cells can also enter G0, a period in the cell cycle in which the cell is neither dividing nor preparing to divide. Meiosis 1 starts with a diploid cell, which can be abbreviated to N. First phase of Meiosis 1 is prophase 1. At the beginning of this phase, homologous chromosomes, which are two sister chromatids, similar in size and placement of genetic information, come together through a process called synapsis. The finished structure of the two homologous chromosomes is called a tetrad. During this process, homologous chromosomes switch segments in a process called crossing over. Crossing over helps create genetic variety. While this is happening, the chromosomes become tightly packed and the mitotic spindle forms. The nuclear envelope breaks apart, and the spindle moves the tetrads toward the middle of the cell. In metaphase 1, the chromosome tetrads, which consist of a pair of homologous chromosomes with sister chromatids, are lined up on the metaphase plate. This alignment is random and ultimately responsible for the genetic variation of gametes produced in meiosis. Microtubules from the spindle attach to the kinetochores of each of the homologous chromosomes. Anaphase 1 is when the homologous chromosomes separate and move toward the poles. However, in contrast to mitosis, the sister chromatids making up the double chromosome remain attached. In telophase 1, the chromosomes are located at opposite poles of the cell. At this point, each pole of the cell has a haploid chromosome set in the duplicate form. Cytokinesis, the splitting of the cytoplasm, occurs concurrently to telophase 1, and the two haploid daughter cells are made. Meiosis 2 is similar to mitosis because the sister chromatids separate instead of the homologous pairs. Meiosis 2 begins with prophase 2. In this phase, the nuclear envelope breaks apart and the mitotic spindle forms. The spindle moves the chromosomes toward the middle of the cell. Next, in metaphase 2, the chromosomes are lined up on the metaphase plate and the microtubules attached to the kinetochores. In anaphase 2, the sister chromatids separate and move towards opposite ends of the cell. In telophase 2, nuclei begin to form around the chromosomes, and cytokinesis, the splitting of the cytoplasm, happens at the same time. By the end of meiosis, there are 4 different haploid daughter cells, each labeled N.